I'm Algis Kalukas, and once again, it's my pleasure to welcome one and all to our free monthly virtual What Talk series. For anyone new, WHAT stands for Waterside Hypotheses, Aquatic Theories. In a nutshell, the fundamental idea goes like this. Chimpanzees and bonobos are closer to us humans genetically than they are to gorillas and orangutans. And yet, of the four great ape species, it is Homo sapiens that is clearly the odd man out. As Elaine Morgan so eloquently put it in her book, The Scars of Evolution, no one is the least bit surprised by this. No one expects that our particular unique traits should be evenly distributed amongst the hominoidae, such that, for example, orangutans were naked and fat, chimpanzees could talk and had big brains, and gorillas were bipedal and relatively good at swimming and diving. No, it is us that have all of these peculiar traits and they none of them. So the argument goes, clearly something fundamentally different must have happened in our ancestors that didn't happen to theirs. And if you're a Darwinist, the challenge is therefore to identify an evolutionary scenario that would have provided the change in the regime of selection to favor all of the traits that make us unique amongst the apes. Now, for most of the last century, the scenario latched upon was, to all intents and purposes, the African savanna. Climate change forced man the mighty hunter down from the trees and out onto the open plains. There, having to scavenge and then hunt, he became bipedal, so he could move more efficiently. He became smarter and acquired language, so he could communicate hunting strategies and tool-making techniques. He lost his body hair, so he could stay cool while running long distances over the grasslands, chasing down antelopes. And yes, the story was largely about him. This is still by far the dominant paradigm in the field of human evolution. For generations, most universities have been teaching it to their students pretty much as a fact. There is, however, a compelling, plausible, and very evidence-based alternative hypothesis. Max Westenhofer in 1942 in German and completely independently, Alistair Hardy in 1960 in English, first published the intriguing idea that perhaps all of these peculiar differences might be explained by a more aquatic past. For years, they were ignored, and their work might have been completely forgotten about had it not been for a fortuitous set of events. In 1967, Desmond Morris mentioned Hardy in his famous book, The Naked Ape, despite most of it promoting the very male-orientated story described above. Luckily, a famous Welsh playwright read it, and while satirizing his general Tarzanist theme, she rescued the more aquatic gem from its pages. Elaine Morgan grabbed hold of the idea and made it her own for the last 41 years of her long and fruitful life, most notably reminding us that women also have played an equal role in human evolution. Elaine's brilliant books on the aquatic ape hypothesis have inspired many further lines of scientific inquiry, some of which have been presented here in this lecture series. This is the 14th talk, and all but one have provided arguments and evidence in favor of at least being open to waterside hypotheses of human evolution. All of the talks have been recorded and put online for posterity. Anyone wants to see them, just go to www.whattalks.com, and anyone who takes the time to listen to them will discover an ever-growing list of smoking guns, indicating a more aquatic past. It is to that list that today we get to add one more, the peculiar phenomenon of vernix caseosa, the layer of cheesy wax which human babies are often born covered with. To tell us all about this, it's a great privilege that we have Tom Brenna live from the USA in Texas. He is Professor of Pediatrics, Chemistry, and Human Nutrition at the University of Texas in Austin. Like Michael Crawford and Stephen Cunane, two of our earlier speakers, Tom is an expert in the biochemistry of fatty acids, and he's been collaborating with them both for many years. I put a potted biography of Tom on the What Talks website, which lists a long and impressive career. But to be honest, he first appeared on my radar when Sir David Attenborough delivered a lovely two-part radio documentary on the BBC entitled The Waterside Ape. In it, Sir David excitedly reports new studies done by Tom and his co-workers on the vernix phenomenon. I've put a link to the documentary on the website as well if you want to hear what Tom had to say way back in 2016, but I'm sure Like me, everyone is now keen to hear what Tom has got to say on the subject today. So without further delay, it now gives me great pleasure to introduce Professor Tom Brenna.
Well, thank you very much, Aldous. It's wonderful to have a chance to share this story, which I think, like many of the stories in this world of the evolution of humans at the shoreline, comes and goes. It seems so having it all in one place is a wonderful thing. So with any luck, you can see my first slide. Good. So the story I'm going to tell you starts with vernix caseosa in humans, and as often is the case with vernix caseosa, the discoveries around it are fortuitous as well. And then it moved a bit into the marine mammal story. So the first thing I'm going to do is tell you about what vernix caseosa is, where it appears in humans, and something about a peculiar composition that it has. That's something of a signature. And then I'll the second part of the talk will be all about the connection to California sea lions and our marine mammal connection. Before I do that, though, I had made this slide a few days ago, having detected a extraordinary paper that came up just a few weeks ago. It was published just a few weeks ago, which was published in Nature, Ecology, and Evolution, called "Evidence for the Cooking of Fish 780,000 Years Ago at the Gesher Benoit Yaakov, Israel." I'm almost certainly mispronounced that, but I put a little map here, look for myself where this is in Israel, and the site appears to, at least according to Google Maps, sit on the Jordan River. And what these folks did was have a look at fish teeth by crystallography, and look at the crystal structure. Now, if the chemists in the audience will recognize that inorganic chemistry, or what's often called synthetic organic chemistry, is an exercise quite different than mixing up chemicals in a vial, but rather is mixing up powders and then sticking them into a furnace or an oven and looking to see what comes out. So this is sort of a kind of a business as usual thing for inorganic chemists to have a look at what they've created. And in this particular case, they had a look at fish teeth and the crystals that existed in these fish teeth. What those crystals tell you is the relative heat treatment, and so the temperature range and the time that the material has undergone. And so I'm just going to refer you to the paper, but I'll just read from the abstract. Aquatic resources were probably a substantial component in the subsistence of early hominids, the proximity of many early and middle Pleistocene, and that would be 2.6 to 0.12 million years ago. Archaeological sites to wetland habitats indicate their major role in homodispersal, and size strain analysis using X-ray powder diffraction provided evidence that these teeth had been, these are fish teeth, had been exposed to low temperature, less than 500 degrees C, suggesting, together with the archaeological and taphonomic data, this is the way fossils are created, that fish from the archaeological horizons of Area B had been cooked and consumed on site. This is another early evidence of cooking by hominids. Well, I think it's more than just cooking by hominids. That's what's interesting, is what they were cooking. So I'll just refer you to that. I won't want to spend any more time on it, but this one is really extraordinary. Perhaps these authors might be enticed to tell us the story in this series. All right, so on to vernix caseosa. I have an outline I've already mentioned. I'll talk about what vernix caseosa is, where it's found, what's strange about it, and the question of whether it's a skin thing or a gut thing, and then the connection to marine mammals. Vernix caseosa as a term translates more or less to cheesy varnish cheesy, and it looks like this, and in newborns, and you see this this white, often described as waxy or cold cream appearing material. It's on it's all over the baby. If you did a Google image search, you would find babies that look like they've been dipped in it. And so you see it in various cracks and crevices and all over the baby. This happens to be Lula, and she was our vernix queen of the grandkids. And vernix's dry weight is about half lipids and about half protein, and it consists of lipidy skin secretions. We're reasonably sure that, and I'll say reasonably only because we can't study humans and there's no other models until perhaps now. It's secreted on the skin, but it also has skin cells sloughed off within it itself. Now, one of the important things about vernix that feeds into my thoughts about what it's really for is that the peak of vernix on the surface of a baby is about 37 weeks of gestation relative to the 40 weeks is a normal gestation. And so what you learn from obstetricians, and if there's any obstetricians on the in the audience, be glad to hear from them, but that post-term babies, that is those who are past 40 weeks, tend to not have very much vernix, if at all. So why would it peak at 37 weeks and then disappear afterwards if it were to A, protect the skin, or B, be fundamentally for enabling the infant to slide through the birth canal? That has never really made any sense to me. So we can talk about that later. But when we first analyzed this material, and I did so because I was asked to do so by neonatologists, that is, doctors who study and treat very tiny preterm babies. I was asked to have a look at vernix for its DHA composition, its omega-3 composition, and we looked at it and we found it had no DHA at all, but it had a whole bunch of fatty acids that we really don't see very much in any kind of internal tissue. And we thought to ourselves, what the heck is all this stuff? Now, without giving you a lesson in gas chromatography, those of you who are used to looking at gas chromatograms will immediately recognize there's a whole lot of peaks in here that you don't normally see. When we did a calculation, we found that almost 30% in a sampling that we had of numerous kids, newborns that is, was about 30% branched fat. Well, you just don't see that anywhere except the skin, and you don't see that in any internal organ. You only see it maybe a bit on the surface. Humans, adult humans, have something like between 3 and 10% of this thing called branched fat, and I'll get to that in just a second. 30% is saturated branched chain fatty acids, or for short, branched fat. So what could this possibly be, and what could it be for? Well, there's a few things that we do know and have known for a long time about vernix, and that is that it appears at the end of the second trimester. So it is more or less a third trimester thing. Might be a little bit either way. It sloughs off as particles starting at the beginning of that third trimester, possibly aided by expelled lung surfactant. That's not important for this discussion. And it makes the amniotic fluid cloudy, and that's a characteristic of a late-term human pregnancy, which is not characteristic of other animals, that there's a cloudiness to the amniotic fluid, which is caused by the particles of vernix that have sloughed off into this sort of sea that the fetus is swimming in. Now, as we learned years ago, the fetus swallows amniotic fluid, and with it, vernix and its branched fat. This has been called by neonatologists and pediatricians of infants the first solid meal of humans. The interesting thing, I don't think I have it on another slide, is that the fetus, as we estimate that the fetus swallows hundreds of milliliters of amniotic fluid every day. That's an awful lot, it would seem. And so it's cycling through. And so what happens to the fluid? Well, it's absorbed in the gut. The electrolytes are absorbed. And it basically is another one of those processes that gets going in utero, presumably in preparation for birth. But what actually disappears is all the liquid that goes into the GI tract, goes into the bloodstream, and then is urinated out again. And that cycle just continues. Now, I have a video because if a picture's worth 1,000 words, a video's worth 1,000,000 words. And this is just a little video that shows a fetus, a late-term fetus. It's a cartoon, that is, late-term fetus and vernix particles being swallowed, and then a little bit about what they seem to do. Not they seem to do, I'm sure they do, in the GI tract. So let's see if this works. Okay, so there's our late-term fetus, and those are our particles of vernix, and floating around in the amniotic fluid. The fetus swallows, and the vernix particles are swept into the GI tract. There is a much smaller concentration of branched fat in breast milk than there is in vernix, but it certainly is there. And that's a space-filling model of a branched fat. And when it gets itself into the neonatal gut, and those are representatives of sort of cartoons of villus, the branched fat basically is taken up, and I'll show you actually data on all this. It's taken up and incorporated into phospholipids where it has a role. And I'll stop right there just for a second. It does have an effect on the gut microbiome. We think perhaps that might be the major role of it, of vernix, that is. It has an effect on the metabolism of the microbiome. So we call it reducing pathogen migration. I'll show you something about that in a moment. And then it enhances a healthy inflammatory response. And again, we have a bit of data on that I will show you. So this video is almost done. Yes, there we are. Okay. All right. So that's it for the Hollywood phase of the talk. What about meconium? So meconium is the funny name for the medical name for the material that accumulates in the fetal rectum during gestation. So everything that gets through the fetal GI tract accumulates at the end of the fetal GI tract, and it represents an integration of all those solids that have passed through unabsorbed, not necessarily unmodified, but unabsorbed. And meconium normally is expelled in the 24 to 36 hours after birth. So we asked the question, are there branch fats in meconium? That is the output. Since there's branch fats in the input, which is vernix, are there branch fats in the output? And then is there a difference in the branch chain fatty acid or branch fat profile between vernix and meconium? Those are two things that we can measure. And so we went to our local hospital, and this is a much later photograph of a graduate student Renat, and let me see if I can get the laser pointer up. Yes, here we are. Okay. And this was her graduate project. And so we went to our local hospital and convinced the neonatologist to sample vernix from the shoulder of healthy term newborns that had vernix, and then also sample for us meconium, and then we extracted and analyzed the branch chain fatty acids. And lo and behold, we found that in vernix, we had quite a wide distribution from 11 carbon fatty acids all the way out to 26 carbon fatty acids. And these are only the branch chain fatty acids. So the branch chain fatty acids have a branch. I think I have a slide coming up that just shows chemical structure for those of you who would like to see that. And so this was a distribution in vernix. The distribution in our meconium was a bit different. The distribution in meconium only had the 16 to 26 carbon branch fat, and the smaller chain ones were missing. Now, you can ask me where they went, but the answer is I don't know where they went. I just know that they were in the input and they were not in the output. And I'll also tell you, just because this can be a bit misleading the way we've represented it, the fatty acids are all normalized to 100% in the particular pool that we looked in. So vernix, all of these open bars normalized to add to 100%, and same with meconium. So it looks like there's much more ISO 16.0 in meconium, but that's not the case. It's just more prominent in the distribution. Okay, so we've established that, and because the branch chain fatty acids differ between these two substances, we immediately realized that they are not metabolically inert. These branch fats are not metabolically inert. So this was way back in 2007, I believe, or that we did this work. I can't even see the date, but it might say 2009 that we finally published it. So what's a branch chain fatty acid or a branch fat? So the chemical structures or representative chemical structures of 3 fatty acids, and I happen to choose an odd chain one to illustrate. The vast majority of the fatty acids internally are linear chains. They're straight chains, and they go even out to 36 carbons, but most fatty acids in humans are from, let's say, 12 carbons out to 26 carbons, and depends on who I talk to as to whether I get an argument about that. But let's say that's the central distribution. So these are linear. Most of them are linear, like you see here, represented as a chemical structure the way you would have seen it in a general chemistry class. The branch chain fatty acids are monomethyls, and so there's a single methyl, and it's at the near the end of the molecule, and it will either be in this configuration or in this configuration. So this looks like it's at the end, but this one looks like it's next to the end, but it's actually at the end, because if you put it there, it would just be this. So this is a branch here, and then this is one branch in, and these are the major ones. In what I showed you previously, there are some with 2 methyls, and there's some with other methyls in other positions, but these are the biggies that we're mostly interested in. Now, as a quick slide that summarizes a lot of work that we've published over the years, I've put the words, I said 30% vernix, and I think that's more like the right number. I guess I made this slide a long time ago. Meconium has something like 13%. So it has a substantial composition missing that lower distribution. Adult sebum is more like 3 to 10%. So that's for adult humans. Human milk has some. Human infant adipose has some, probably coming from vernix. That's my guess. U.S. retail cow's milk, we've done a careful study. We published a careful study, and about 2% of retail cow's milk is branch chain, and that almost certainly comes from the rumen organisms. And we also did a calculation showing that the average U.S. adult diet has something like a 0.5 gram per day of branch chain fatty acids. Now, that's really significant because that is quite a bit more than is in the average American diet for DHA or EPA, the omega-3s. DHA, the last number I saw was 70 milligrams a day. So this is branch fat at 500 milligrams a day. I've never quite understood why people didn't study this more, but because of its prominence in the diet. But that's the reality. Now, where do we think it's important? Now, bacteria, it's certainly important. And how do you count species of bacteria? What are all the species? Well, my eyes fall on to review articles, and so I estimate that it's important in, I don't know, 15 to 20% of species. There probably is some way of counting them. That number would be more or be less, but the point is it's not a trivial number. And particularly in bacillus species, and some species have greater than 80% of all their fatty acids as branch chain. Now, what did they do in a membrane? Well, they do something, I will call it rather clever. And I've never been a big fan of the concept of membrane fluidity as it relates to the very long chain fatty acids and unsaturated fatty acids, but clearly it's doing that. These branch chain fatty acids are regulating the membrane fluidity and the membrane properties of these membranes. They certainly reduce the melting point and the membrane transition temperature. But importantly, unlike unsaturated fatty acids, branch fats are not susceptible to oxidation. So rather than putting a cis double bond into the chain, and that double bond being susceptible to oxidation, it is not susceptible because they're saturated fats, as you can see here. And so that is almost certainly the reason why they are mostly surface lipids. And when I say surface, I mean skin and other surface regions that are maybe even more specialized. And I'm going to go quick and just say that this review from 1991 still may be the best one, and looked at branch chain fatty acids in bacteria. And I pull out just a couple of sets of organisms from a table there. And here's the bacillus species, and these are the percentages of branch fat in these bacillus species. And you can see the numbers are 80%, 94%. They're all quite high numbers. So these are not rare fatty acids, and they're quite prominent, and they're synthesized de novo by these bacteria, which explains why the rumen bacteria synthesize them and then they end up in milk. Well, so what about biological function of these branch chain fatty acids? And I didn't say it, and I should say it before I go into this. To the best of our knowledge, vernix caseosa is a substance which is unique to humans. It does not appear in any other species. There is one very old report of vernix in a chimpanzee. I've seen amniotic fluid, though I never analyzed for it. I've seen amniotic fluid from newborn baboons. It's not cloudy, and you sample surface lipids from newborn pigs, newborn dogs, and you don't see it. So we don't see it in other terrestrial animals until the study that we did here, not this particular study, but our work, which I'll hit in just a few minutes. The only species known to have a true vernix was, and accepted to have true vernix, was humans. Okay, so what does that mean? Among other things, that means that there's no good animal model because no animal has vernix as that would model, let's say, prematurity. So I told you that that vernix begins to develop at about week 27 of gestation. Some people say earlier than that, but certainly not much earlier than that. Certainly not before halfway through gestation. And so if it has something to do, if vernix, that is, has something to do with the health of, let's say, the GI tract, let's make that as a hypothesis, then we might imagine that there would be a condition in humans that would be associated with the absence of branch fat. And it turns out that there is a condition in humans where there is an absence of branch fat, and that is very low birth weight infants that are born at, let's say, earlier than 27 weeks gestation, and that condition is known as necrotizing enterocolitis. Necrotizing enterocolitis is a devastating disease of the intestine in premature infants. It spontaneously occurs in something like 7%, but the smallest kids have a higher percentage. And it causes, of that 7%, about half of them die. Now, there's been a lot of progress since we started doing this work on what might be causing it, but it still is a bit of a mysterious condition in terms of the underlying causes. So in this study that we did not long after that original work, I think this was published, what do I have here? 2011, we used a neonatal rat model of prematurity. There are a couple of different models that one can use, but as I told you, rats don't have vernix. So we wanted to see whether branch fat would have an influence on the development of the necrotizing enterocolitis-like condition. And to make a long story short, I'll just refer to two bars here. Here are essentially the normal controls. And when we gave the premature rat pups branch chain fatty acids, substituting for what amounted to the normal feed, we reduced that incidence by about 50%, a little bit more than 50%. That was really a first try. Not much has been done on this since, which makes me cry a little bit, but it still is out there, and it still is was very, very kind of clear effect. Okay, so in the context of that study, we had a look at the microbiota, the distribution of microorganisms, and we did it a little bit differently than most people do microbiota. We had it analyzed, but then instead of doing the usual the usual principal component and analysis and the usual kinds of generic analyses that are done to try to put all of the organisms in one place, we looked at the levels of some of the organisms that rely on branch fat in their membranes. And what we found is that in the pups that got branch fat, that two organisms, Bacillus subtilis and then Pseudomonas aeruginosa, both had much higher amounts of, were much more abundant in the branch fat treated animals than the ones that were not treated with branch fat. So in other words, makes sense, organisms that use branch fats in their membranes were at advantage. They didn't have to synthesize them. They could take them up from the environment and their growth was accelerated. So that's rather important. I'll also point out that the healthy animals, that is, we take all the animals and regardless of group and refer to the ones that are healthy versus the one that had necrotizing enterocolitis, that the Bacillus subtilis particularly was much higher in the healthy animals than it was in the neck animals. So we're doing something to the microbiome, and again, I can get into a lot of detail on that. Now, some of you might have picked up that I had mentioned Pseudomonas aeruginosa, which is really a pretty bad pathogen. It's thought of as a killer in the neonatal nursery because it can become a pathological infection and take off. Now, Pseudomonas is a fairly ubiquitous organism. So one mystery is, why is it that it kills some kids and doesn't kill others? Now, this is a study that was done by someone else in Japan in 2008. And I'll just go through this very quickly. This is a motility assay. So these are Pseudomonas, and you put a dot of Pseudomonas in the middle, and then you incubate them for a while, and they crawl around and they expand out, and crawl around. And if you put various fatty acids on them, and these are the conventional fatty acids, let's say 14-0, 16-0, 18-0, and then a couple of shorter chain isos, anti-iso and iso, branch chain fatty acids, they still crawl around. But if you use a distribution sort of in the middle of chain lengths, 15, 16, 17, Pseudomonas does not crawl around. So Pseudomonas just sits down and is quite happy right in the middle there. So this motility is related to the pathogenicity or the virulence of the organism. And so what this shows at minimum is that these branch chain fatty acids have an effect on the metabolism and the function of a pathogen, Pseudomonas aeruginosa, and almost certainly from an assay like this, one would conclude it makes it less pathogenic. So that's an important effect as well. So let me keep going because time is passing. And I'm going to do this slide very quickly. These were human fetal cells in culture, and these human fetal cells were treated in our lab with branch fat. And we found remarkably that the cells take up and will incorporate, we treat them with free fatty acid, but then we look for phospholipids, that is the lipids in the membranes, and we find that they plug them right into the membrane. And at levels that are even exceed 60%, depending on the particular branch fat. And the cells seem pretty happy with them. They're not dying and floating and all kind of stuff. So two things seem to happen with these branch fats. One is that they alter the microbiota seemingly in a positive way. And secondly, they alter the enterocytes, that is the cells of the baby that are lining the guts, in a positive way. And in other studies I don't have time to get into, we show that it modulates the inflammatory response and tamps it down a bit, actually, so that it doesn't go crazy as one would expect in necrotizing enterocolitis. Okay, so now I get on to the marine mammal part, and that first part was really just sets the stage for all this. And I sometime in the 1990s came under the spell of a couple of guys in the picture here, Michael and Stephen, who I last I looked got on. So hi guys, that's a photo from Michael's backyard and quite a few years ago now, and we've continued to work together, and this is actually a pretty good paper. Would be glad to pass along if anyone's interested. We're still talking about DHA and affecting and improving the brain and all that kind of thing. And this is of course me pre grandpa mustache. So so let me tell you the genesis of this idea that we sort of got into this. So around 2005, and I was unable to find in a quick search the other day the exact documentary, but it was a David Attenborough documentary around 2005 where he interviewed Don Bowen, a marine biologist, and I think perhaps known to some of the folks who were interested in the shoreline hypothesis of human origins. And I saw this not in 2005, I saw it much later, and in 2009, and we had already done this vernix work, the original vernix work, and published it by that time. I wrote, and I found these emails the other day. I'm writing to inquire about a statement attributed to you in a David Attenborough BBC presentation. Vernix is found on some species of newborn seals. Remember, I was unable to find vernix on any terrestrial mammal, so no model. And I searched for a scholarly paper, didn't find one, and wondered if he could find a reference. He wrote me back immediately, and he said, unfortunately, there's no dedicated study of this in harbour seals. That's who he was looking at the time. This is my bold, not his. Vernix was not the subject of our work, and we admittedly paid very little attention to the waxy substance covering harbour seal newborns and did not report it in our papers, etc. So that got me interested in this, and I thought, all right, I've got to find myself some sea lions, and then of course, as an experimental scientist, you start thinking, well, gee, this could be a model. And I had this image of a very large, let's say, aquarium that could hold some of my experimental animals. But of course, I rejected that pretty quick. I'd have to take over a SeaWorld, I suppose, to do something like that. Well, it was just a few years later that, and this was, I was at Cornell at the time. I was there for 28 years, so and still am there to some extent. And Cornell has quite a well-known veterinary college. It was the first in the U.S., and someone was giving a seminar on marine mammals, and so the graduate student whose photo you saw earlier sent her over there. And she met, that was the beginning of our relationship with Judy St. Leger, who's an aquatic animal veterinarian. And she was our hero because she got really interested in this whole question of ours. At the time, she was a research veterinarian working with SeaWorld, which is a chain of amusement parks based on aquatic mammals, among others. And so to cut to the important sort of part of the story, she also was very well acquainted with the marine mammal centers in California, and she was on the lookout for samples for us. And it turned out that I don't know, I hate to say it, but it seems like God gave us some samples. So there was an algal bloom that caused a poisoning event, and there were deaths in numerous pregnant California sea lions that washed up on the shore. So all of these animals are necropsied, and she managed to get us tissues and fluids acquired from those necropsies. So first, here's a photograph, and so now we have a photograph of we were first saying skin debris because we're trying to be cautious, but vernix on a near-term seal. So there is the particles of vernix, and I've reproduced the photograph you saw earlier of the newborn that has vernix on her, and you can see right here, and I'm intrigued by all this vernix that's right at the mouth, which is what you would expect it'd be collecting on these whiskers if this animal is swallowing this stuff, which I'm quite sure that they would have every reason to believe that these animals in utero would be swallowing vernix just like human babies. So, okay. So, of course, you can't take samples of lots of things in a human baby, but you can in a dead seal. So we looked at, we had samples of that vernix, that surface material, sample of amniotic fluid, sample of stomach contents, sample of meconium, and a sample of blood serum from these animals. Here are branch fat concentrations along here, and these numbers for vernix, the surface lipid, amniotic fluid, and the stomach contents were actually a bit lower. And these are individual animals. I should really point over here because this is the mean of 6 animals. And so here is, again, vernix, amniotic fluid, stomach contents, meconium. Look at meconium. That means the branch fat is being swallowed and making it all the way through, just like in human babies. And then serum is quite low because not much gets into serum, but that's what we get. So we found not percentages of 30%, but we found substantial percentages that we would not have expected if this was not a similar phenomena. The amniotic fluid was quite high, almost 10%. Big error bars, but these are incidental data. And again, I remind you what the human data are. We published 29%, 17%, but these numbers are quite high. And here were actual samples that we got from animals that were born at the Cornell Vet School. We had a couple of litters. Dog was at 1%. This is in meconium, and piglet was at 0.12%. These numbers are obviously very low. And so on this basis and the basis of the appearance and the rest of it, we believe we've got true vernix on the surface of these animals. And here's another thing, and I'll mention 2 things: branch fat and squalene, and I'll tell you what squalene is towards the end, and I just have a little bit more to go. we asked the question, what is the concentration of the amniotic fluid branch chain fatty acids that is as an indicator of vernix sloughing off into the amniotic fluid as a function of fetal weight. Remember, we can't do gestational age. We can only weigh the animal. We could estimate gestational age from the weight, but that doesn't get us anywhere. We just do the weight. So when we look as a function of weight, it's very low, as you would expect in humans. Not as you expect, it's very low like it is in humans, and it comes up as it does in humans to a plateau. And this is in meconium. So that's amniotic fluid, that's meconium, same story. So it's going from the amniotic fluid to the meconium, same story. I will point out that squalene, which I'll get to in a minute, is more linear as a function of weight, so a little bit different. It's actually quite a bit of squalene in these animals, but at any rate, this was, again, one of the other, while we're talking about smoking guns, the patterns that tell us that this is working the same way as it does in humans. So what function might be served by vernix in the newborn gut? Well, and why is it more important to have vernix for humans and for sea lions and maybe for other aquatic mammals? And I would make the point that the data is more pointing to aquatic animals that are born on the shoreline rather than whales and porpoises that would be born in the ocean. The speculation, more or less, is that I'll say more or less. We'll just go with the speculation is animals born on the seashore may be vulnerable to ingesting pathogens by swallowing environmental water before nursing. If you take a look at most animals, they seem to be born and then go straight to the nipple. And I used to wonder as a kid why it worked that way. You'd think that. I don't know what anyone else would think. I would have thought that they would be reasonably well nourished as they were just born and only go later. Well, they do seem to go to the nipple as one of the first things they do. Well, why do they do that? It may well be a microbiotic, gut protective kind of behavior. And so if one of these guys being born on the shore gets a slug of water, that could be devastating to the GI tract before mother's milk arrives. And so we speculate that vernix may protect the newborn GI, that is the gut, the newborn gastrointestinal tract, as the first bacteria are arriving to colonize the gut. And as I showed, at least in our cell systems, it seems to moderate inflammation and favor these commensal organisms. But if someone put a gun to my head and said, "Are you sure this is right?" I would have to say, there's only one way to really understand this, and that's to do experiments. We can't do experiments with sea lions, so we can't say 100% for sure. Okay, I just got a couple more slides, and then I'll be done. The story of squalene. A person that I was in close contact with at the time we were doing this said, "Why don't you take a look at squalene?" And squalene seems to be a rare compound, a compound that's rare in the animal world. So I started digging a bit, and I found a paper back from 1980 where a group in Iowa, dermatologists in Iowa and colleagues had analyzed the surface lipids, the skin lipids from 46 different species. And there's a lot of molecules you can't measure very well in 1980. Well, squalene was one you can, because you can use thin layer chromatography, and that's a perfectly reasonable approach. And when I dug through my archives to make this slide a few days ago, I refound the paper. And for those of you who are used to looking at thin layer chromatography data, what we see are these bands that are indicative of various molecules, and I'm showing you 1, 2, 3, and squalene bars, and that is the squalene standard. So that tells us that those 3 are in fact squalene. And I later found another paper that they had looked at, that they had published a few years later on a particular species of, I think it was moles. Yes, moles. And in that paper, they make this statement: discovery of a major proportion of squalene in the sebum, that's the surface lipid, of this animal extends the number of non-human species that have this characteristic to 4: otter, beaver, kinkajou, and then the mole, all of which inhabit damp environments. Kinkajou lives in the rainforest, suggesting that squalene conveys some biological advantage under these conditions. Now, I didn't say it, but I should have. Human sebum has squalene. It's actually quite prominent in human sebum. So we have something in common with these animals that live in a aquatic environment or a wet environment, some wetter than others. And so you can put a picture of a human on here. And then we went and looked, and we found the same, we found the same thing in, we found squalene in the surface lipids that vernix, and I showed you a couple slides earlier, that are in California sea lions. So now we have 6 species. So you can throw sea lions in with humans as having squalene. So what's squalene? Some of you can bring it to mind. This is squalene, the way it's often drawn. It's an intermediate in cholesterol synthesis. And so it doesn't look like it, but the organic chemists in the crowd will immediately recognize what's known as an isoprenoid. So it's a polymer of a small molecule called isoprene. If you take this molecule and you bend it around a little bit, you will find it makes a nice shape that can be synthesized by enzymes into this molecule, and then finally into cholesterol. And so this is a cholesterol precursor, and something like 13% of human sebum is squalene, and that is a heck of a lot, guys. That is a lot of stuff on the surface that's that squalene would represent. When I say 13, I mean 13% of I guess that would be total mass. It might be 30% of lipids. Most of it is lipids anyway. Now, the other thing the chemists will notice immediately is that this is a very hydrophobic, that is water repellent material. And it's known that the surface of the skin has polymers that are made from unsaturated fatty acids. And I really am beginning to wonder, and for all I know, this is already known, whether squalene with its double bonds participates in that polymerization, further making a water barrier. So I'm almost done. I do want to make a special acknowledgement in much of this work that we've, at least our thoughts, some of the thoughts that have wound around in the marine mammal story. I got myself connected to Richard Fox Collins, who was was a writer for David Attenborough, and David Attenborough wrote me this. This is actually sitting in my place. I was inquiring as to where the origin of the idea for vernix being related to this story, and Sir David writes, I am quite sure that the suggestion that crucial evidence might be found in the, and I can't read that word, in the matter of the vernix was first suggested to me by Richard Collins, signed David Attenborough. So I also got Sir David's autograph, which wasn't bad. But at any rate, Richard, who's a co-author on the seal paper, and that photo was from a memorable visit the two of us made to Bletchley Park. Really seeded many of the ideas as we went through this. So I'm at the end here. 2 species, humans and California sea lions, make a lipidy material, I will call it vernix caseosa, in utero, that enters the GI tract. Vernix is rich in branch fat and squalene, both of which have established biological roles. Don't know what the squalene might be doing in the neonatal gut, but maybe that's another question we'll look at sometime in the future. And then I leave you with the smoking gun question: what do humans and California sea lions, and maybe that other list of beavers and otters and kinkajous and moles, have in common that humans and most other terrestrial animals do not? And maybe it has something to do with origins. So that's my story, and thanks so much for your attention.
Well, thank you very much. Thank you very much, Tom. That's brilliant, really brilliant. Some lovely slides. I love the videos, and it's kind of stretched my mind a little bit, going back to some of the biochemistry there. But very, very fascinating stuff. Now, I'm very aware that we've got quite a few people, so I'd like to open it up for questions. I've got one to ask. I've got lots of questions I'd like to ask, but I'm just going to ask one just while people are thinking of putting in their questions. So if you want to put your hand up, or if you want to go into the chat and post an intention to ask a question, I'll try and manage it so that everyone gets a fair go. So my question really is, I'm really interested in I think you said earlier on in the talk something about chimpanzees having vernix, and maybe there was one study or something about that. I just wondered if you could maybe elaborate on that, because that to me is always a very fascinating area. It's going back to this point about ape-human differences, and is it really a unique thing about humans, or is it something that the great apes might all share with us? Just maybe if you could elaborate on that point.
I'm glad to do so. When I was really up to my armpits in this story, I was unable to find any clear data, and it may exist and I may have missed it, but I was unable to find much on vernix in primates. Now, I did a lot of work prior to all my vernix work in non-human primates, in baboons and a bit in rhesus monkeys, and they were also newborns. And it's one of those things that you say to yourself, gee, if I knew this was going to be a question, I had the perfect model. I could have sampled amniotic fluid and other things, but I didn't. So I could have answered this question analytically, but the reason I brought up the chimpanzee paper, and I actually have a slide on this, but I won't bring it up because it's a paper that comes up from time to time. People say, oh, well, somebody published vernix, as somebody said vernix was on a chimpanzee. Well, it comes from a single paper in 1937 from Yale, where there is one sentence that refers to some white debris on a surface. Well, all white debris on a surface is not vernix, and the reason why we went through all that work to acquire samples is to is to actually do the chemical analysis so we could get some idea of whether it really did resemble vernix. So the only point I'm making is that this old paper from 1937 to me is not evidence at all that chimpanzees have vernix, and I know of no evidence that non-human primates do. You will remember and you'll recall that non-human primates are born skinny. Stephen Kinane has this stuff off the top of his head, and I can tell you a newborn baboon looks like an old man. They have no fat on them, and so they're quite different from that perspective. But I can't say too much more about whether anyone has really looked carefully as to whether non-human primates, the great apes, have true vernix. Just don't think the study's been done, but I'd be glad to be corrected if anyone knows of a paper.
Great, thank you very much, Tom. That's really great. Now, just before I pass on to anyone else, I was wondering if would it be okay to put a link to your paper? I think it's Wang et al. The paper that I think you did about the harbour seal. Could I put that on the website? Is that okay?
You could put links to all our papers on the website. The more the merrier.
Okay, great. Okay, so who wants to go first? Anyone got a question? Leslie's got a hand up. I'm trying not to be biased here, but okay, Leslie, you go first.
Thanks very much, Tom. That was absolutely fascinating. One question that I've got is just, do branch chain fatty acids actually provide any nutritional benefit for the growing fetus in utero?
Yes, excellent question. Because these molecules are not very well studied, and I sort of alluded to that, Even though they are substantial, I'm not sure that anyone really knows what nutritional benefits they might be other than in gut health. And so I would cite the two things that I already have, which is one is that they modulate microbiota, and the other is they almost certainly modulate the properties of the enterocytes, the cells lining the gut. But the other thing that they almost certainly do, but I don't know it's ever been shown, is that they are almost certainly transported to the skin, because we have branch chain fatty acids in the skin secretions, the sebum secretions. So when I've spoken a little bit to dermatologists, they sometimes talk about beauty from the inside out rather than cosmetics. Maybe we eat something and then it goes inside to inside out. And so the stunningly obvious place where they would go would be the skin. So they probably support skin somehow.
Fantastic, thank you. Just one other funny little thing. I'm a midwife, and midwives like to keep the vernix on babies for a good 24 hours after the birth. And midwives also like rubbing it into their own hands because it has really lovely qualities, which isn't very hygienic, but there we go. Can you see any benefit to leaving it on the baby for any length of time after the birth?
You know, that is exactly the question that I would ask you. And again, it's so difficult to be mysterious about this and say I don't really know, but there are no models, and the way we get answers to questions about whole animal kind of things, how does the whole skin work, is by animal models. And the skin is remarkably, skin lipids particularly, are remarkably poorly modeled by the usual, well, these days, mice that we typically study. So, but I don't see anything wrong with it. So I'll go that far.
Brilliant. Thank you very much.
Who would like to go next? Francesca, and then Stephen, please.
I was just curious if what peer reactions were to your work, and whether it had created any sort of response, or if anyone had come up against it in some way, thinking that it was promoting aquatic theories.
Thank you for the question. I do not recall any serious place where we were attacked. We were, because our story was very, let's call it analytical, in the sense that we were sampling in a certain way, analyzing in a certain way, and presenting those data. And we didn't put a title that says, aha, we have it now. We obviously evolved on the seashore. There was not a lot of feedback. There was not a lot of attacks. And when Sir David's BBC Radio 4 piece came out in 2016, we didn't hear a lot. That's where I almost would have expected to hear more, where it was being used in support of this crackpot, and I'm referring to myself here, theory about shore-based evolution, and so I don't know that there was a heck of a lot of that. I mean, you can always find something on the web where someone says, well, Sir David shouldn't be talking about this, because we all know it doesn't work that way. So occasionally there are things like that, but there wasn't a lot of attack on it.
Okay, thank you.
Did you want to come back on that, Francesca?
No, not that sounds fair enough. I want, maybe it's the case of, like most smoking guns, when it comes to this hypothesis, they do what they usually do, which is kind of ignore it.
Well, I think that's probably right. I mean, it's like a lot of things where you say, okay, well, everybody's looking around at everyone else thinking, do I have to deal with this, or will it just pass, and then we can keep saying what we've been saying all this time. So
yeah, I mean, on that documentary, David Attenborough, Alice Robertson, Mark Maslin did write a very critical piece on the web. I mean, it was just a web piece. It wasn't a peer-reviewed paper, but it was still pretty horrible and damning. Stephen, do you want to go next?
Sure. Well, congratulations, Tom. It was a stimulating ride to go back over that material, which I'd never really heard you speak about much before. So it was a pleasure to hear it again. I've two brief comments and one question. One is perhaps in response to Leslie's question about it, what's it doing there? And I wonder whether it has any, obviously protecting against dehydration across the skin. Is that ridiculous, or is that actually plausible? That's one comment. The other comment is, I think you mentioned it peaking in utero at around 37 weeks. And I don't know what the time course of the development is, but it sort of reminded me of adipose tissue on the fetus, which is certainly taking off exponentially at that point in fetal development. So maybe that's a coincidence, maybe it's not. And the question is, I think you said somewhere near the beginning that you didn't expect it to have much relationship with lung surfactant, or some reference to lung surfactant, that you sort of were separating the roles. And I would have assumed that some of this material is getting into the lungs. We want to protect the lungs as well against infectious agents. And so, and of course, it's full of a lot of saturated fatty acids, surfactant. That's primarily 16-palmitic acid is a major component. So is that a coincidence, or is that something that maybe would be instructive to study?
Thanks for that, Stephen. The first one you asked about is what role does it have on the skin? And I've been reluctant to say, gee, I know that it's there for hydration, because if it is there for hydration, then how come it goes down in normal babies when they're born at the normal term babies when they're born at 40 weeks? Number one. Number two, why haven't other animals developed it in some sense of convergent evolution or something along those lines? And it can be quite variable. So every baby doesn't synthesize the same amount. So the reason I'm reluctant to again, that's really the reason why I'm reluctant to make too many comments about it. Oh, and there's a fourth reason. The fourth reason is that, as far as I can tell, everybody in the literature is studying the skin properties insofar as they do study it. And so there's plenty written about the skin properties, at least by the neonatologists. And the story of leaving it on the baby as long as possible, don't towel it off, has been around for, I don't know, at least 10 years. It seems reasonable to me. So that's the first question. The second question is, the adipose is taking off at 37 weeks. Okay. The adipose, and you know what those curves look like. The other thing I was thinking with regard to adipose, or at least not adipose, I take it back, with regard to the increase in, let's say I'll refer to DHA. It was a few years ago that the Dutch group, and I'm blocking on the name early on my Sunday morning, it looked at the accumulation of, let's say, DHA, and it seemed like half the DHA accumulates in human infants after 35 weeks. So the first 35 weeks get you one half of it, and the next 5 weeks get you the other half. And and so this whole business of fat, another one of those compelling things about fat being important in a bunch of ways for humans. Now, a perfectly reasonable human baby might be born at 35 weeks, but they do adipose, they do DHA in the brain, and they get the peak of vernix in those last weeks of gestation. So there's something going on there. I don't know whether the same phenomena is underlying biochemistry is at work in the adipose and in the skin. Last thing, lungs and surfactant. Years ago, one of the groups that was studying vernix recognized that the fetal lungs are, let's say, breathing amniotic fluid. They are the fetus is breathing, in the sense of the lungs and the diaphragm moving moving amniotic fluid in and out of the lungs at something resembling a respiration rate. I can't tell you what it is and whether it's fast like a little kid, but it's doing that. One of the bits of speculation back then was that the surfactant that was produced in the lungs was enhancing the sloughing off of vernix from the surface of the baby. And so I was more just referring obliquely to that. We've analyzed our share of surfactant lipids and our share of lungs, and haven't seen much for branched chain fatty acids in lung lipids, though you are, of course, quite right that they are very saturated, like, but most of that's palmitic acid, a little bit of oleic acid, a little bit of stearic acid, but there's not much branched fat in them, and so I conclude from all that while I haven't excluded an effect, I don't see one yet.
Okay. Do you want to come back on that, Stephen? Is there anything you want to add? Or
Oh, that's good. No problem.
Great, thank you. Well, just before open up to anyone else, I've noticed here we've got Michael Crawford. We've got the three gurus of DHA world here, and I just wondered if Michael wanted to say something or contribute.
Michael's microphone isn't working, but he's put a question in chat.
Oh, okay, right, okay. So let me read that. Let me read his question out. Michael says, "Hi, Tom, brilliant, just brilliant. My microphone doesn't seem to be working, but I have a question. You referred to a branched chain fatty acids linked to liquidity. We know membrane oleic rises if arachidonic and DHA fall. You referred to the branched chains linked to birth weight. My question is there a relationship between membrane branched chain with membrane arachidonic and DHA?" I hope I said that correctly.
You did well. You did well, and you and you would think I would know that off the top of my head. So here's a couple of points that I'll make. The first thing is you don't generally find DHA and arachidonic in the same places that you find the branch chains. So you don't see much. So I alluded to vernix having 0 DHA. That's what I was first asked. How much DHA is there? The hypothesis was that maybe this vernix that was being swallowed might be a source of DHA. Maybe DHA being made on the skin, and that it would be, let's say, a source of DHA. That'd be pretty cool if it worked that way, but it doesn't work that way because there's no DHA in vernix. In microorganisms, they tend to be very unsaturated. They tend to be very saturated if they've got branch fat, if they've got branch chain fatty acids. Now, I would have to be thinking about a situation, as you've put it, where both branch chain fatty acids and DHA, EPA, arachidonic, do they appear in the same place, in the same lipid pools? And I'm having trouble right now even thinking about where that might be. It could be in the gut lipids, of course, because there's a DHA and arachidonic in the gut lipids and the enterocytes and all that sort of stuff. So there may be something to that, but I'm hard pressed to say too much about that. Yeah, sorry I let you down. I'll go dig a little bit and you get me thinking as usual.
Oh, that's good. Stephen, do you want to chip in?
Yeah, I didn't have anything to contribute on the DHA side, but it struck me that I didn't appreciate, Tom, from your talk, where things start. What is the origin of the branched chain fatty acids? And there seems to be a cycle between the skin and the gut and the amniotic fluid. Do you have any sense of where things start? Is the mother actually producing these branched chain fatty acids in the placenta or somewhere? Or where do things start, in fact?
Yeah, that's an excellent question. The way we often answer those questions is by finding an animal model and studying it in the animal model. So Well, yeah, no luck unless we want to make an experimental colony of sea lions, which already I think we might have a problem with. So that's the first answer that I'll give you. So then the second answer I'll give you is that the underlying biochemistry is reasonably accepted, let's say, and that is the branch chain amino acids are I guess it would be valine and is it leucine or probably isoleucine are deaminated and then seed fatty acid synthase, which then makes these molecules. So that underlying biochemistry is in okay shape. That doesn't answer your question. Are they made in the sebocytes? That's where you guess that they would be made, in the sebocytes of humans. And that's what the presumption is, that they're made in the sebocytes. And I think they are made in the sebocytes. I think there's a bit known about that. Human cell culture in skin cells, at least for some of them, is a little bit. So I remember last I looked at this was not as didn't have as much fidelity to the in vivo situation as one maybe would like. So the presumption is that it is on the that is in this it's made in the skin and not made by the mother. It's not made by the mother. That's the presumption. That seems to be the case. But I can't really exclude it. I mean, we can't exclude it. We can't say, well, the mother doesn't transfer any from the placenta into the through the amniotic fluid. I mean, I guess that's possible.
Well, there's an interesting coincidence that you bring up branch chain amino acids, because they're also ketogenic.
Wonderful.
And that's an interesting parallel between these two stories that hasn't occurred to me until now. But they're relatively efficient ketogenic substrates as well.
Are they?
So
okay.
we might be doing two birds, one stone.
We may, and you've pointed out that ketosis seems to be important for
brain energy metabolism.
Yeah, brain energy metabolism. Well, not just that, but you've also pointed out that for infants, it seems to be important to top off the energy and the fuel.
And the lipid synthesis, though. There's more ketones. There's more ketones derived from from alpha linolenic acid going into cholesterol, which you talked about, than going into DHA. And so it's probably again a potentially common route between the vernix and brain lipids. In fact,
I hadn't thought about this until this conversation. Well, it's good to put things down and pick them up again. So thanks for the chance to think about this again.
Fascinating stuff. So Francesca, do you want to go? And does anybody else want to raise a question?
Yeah,
or
just
make a point. Francesca, go on.
Yeah, you mentioned that the California sea lions, you had those samples because of an algal bloom and a toxic event. Is there any other, or it's just not possible to know? I mean, can you rule out, or does anybody know anything about the fully aquatic mammals, dolphins and whales? Is there any way of finding out whether they have vernix, or it's just not knowable at this point?
Well, I regret to say that Judy St. Ledger, my sampling hero, retired not too long after she retired from SeaWorld, so didn't have so much access to those aquatic animals. And so I remember she really very intriguingly told me that she thought that maybe baby walruses had a lot of vernix, and we ought to. And so my latest email was I was asking her again if she could find me a baby walrus, but I don't think at least in the wild I would want to be tangling with the mothers to try to scrape a little vernix off of them. So and I haven't been able to find even a video on YouTube that was good enough to really say one way or the other. So that's my way of responding to you, that I have had certainly the thoughts about continuing on and doing something along these lines. And for all I know, now I'm speculating even more, but for all I know, a whale or a porpoise needs all the more sort of content of the gut before it can go nurse and start. Yeah. For all I know, that's the way it really works. And but again, how do you do these studies? It's non-trivial. So I'd love to go ask the NIH for money for this, but I think I will be wasting 3 months if I write a proposal like that. So maybe the NSF. I don't know. Good thinking.
Because it would be, I mean, it would be amazing if it turned out that all semi-aquatic shoreline giving birth on the shoreline mammals had vernix on their pups, and even fully aquatic mammals, if anyone ever could go that far.
Yes, I feel that suggestion making its way into the same brain cell that that the sea lion thing went. So so thank you for that. I'll give you a talk on that in 5 years.
Well, it seemed fascinating to me, Tom, that you mentioned the hypothesis that it was the shoreline dwelling mammals that might be particularly vulnerable to getting a slug of seawater, which somehow would contain some sort of pathogens, that bacteria presumably, that would cause an infection. It seems a very specific sort of problem to have a very general solution to, though. That's my problem with all of this. I just think, why only in shore-based mammals would this be required? There's a particular colony of bacteria that we know of that would make a newborn vulnerable to an infection that the vernix would protect against. Is that the hypothesis?
The bottom of my slide said I don't really know, and this is where it leans right into that. Those are exactly the kinds of things one would want to know the answer to, but there really has been no follow-up on that. We just don't know. So it's kind of a okay, well, maybe we'll look over there someplace, and that's the best I can do.
Fascinating area, though, isn't it? Plenty of research to be done on this. Stephen, you had your hand up a second ago, and then anybody else would like to go after Stephen?
Yeah, thanks. I'll just the image you showed us of the fetus, the harbor seal or sea lion. I don't recall, but there was, after they washed up dead, and you were able to collect samples. Are they hairless at that point, those fetuses, or is there actually the hair cells are present, or the on the skin? I'm just wondering whether is there something exclusive about the development of vernix in a relatively hairless species like humans versus non-human primates and terrestrial mammals? Is there some kind of exclusion that we can make along those lines, or is that completely false?
It's a good question. I'll just point out that those were those seals those are California sea lions. They were in utero. So it was the pregnant animals that washed up on the beach, and then when they did the necropsy, that was a photograph of the animal that was in utero. They look to me like they've got hair on them, but I never really looked that carefully, so I probably will do so after. But I don't know that it's a hairless thing. One of the reasons we went and checked pigs at Cornell for the branched chain fatty acids in the meconium is that we thought, okay, well, they are hairless, and we didn't find branched chain fatty acids in their meconium. So that was precisely the reasoning on that.
Right. Okay, well, I'm a little bit conscious of the time. We're getting close to an hour and a half. So has anyone else got any points they would like to ask or raise? Okay, well, in that case, I think I'm going to close it down. So thank you again very much, Tom. That's brilliant. And thanks everyone for coming and participating. I'm sorry if there was a bit of a confusion over the timing of the talk, because I've been doing this crazy traveling, and when I actually put the invite out, I'm not sure what time zone I was even in. So if there was any problems, I apologize. Hopefully everything was everyone got here anyway. So thank you very much, Tom. Well done. Brilliant.
Thank you.
Thanks, everybody. Take care.
Thank you.
Thank you.
Bye.